Today, I'm taking another play and we're talking about how the structure and plans of this org can best facilitate patient perspectives and community needs. And this is something that is near and dear to my heart because we find ourselves in a very interesting position. Several years ago, we just wanted any opportunity to be heard, to have experiments, to have any kind of data about our disorder. And over the last couple of years, what we found ourselves in is an abundance of opportunities because the science has progressed and we have progressed with clinical assessments and we've gotten more community feedback. And so what we want to talk about are what those opportunities are and how we can leverage them. So to start, let's look at where we are now. We have over 500 families in our network. And I want you to imagine, think about all of the people that you see around you. Imagine 10 more of those groups sitting side by side with you. Imagine how much our community has grown. We're represented in over 40 countries, and that's growing. And not only are we represented in countries, but families are taking things into their own hands in these countries and finding local researchers that can accelerate research. And now we're plugging back into that, and we're really leveraging a network that spans the world and all of its resources. We have 140 research network members, and I do just want to give another shout out to Dr. Dominique Lessard because it couldn't have happened without her. And these network members aren't just calling it in. I think what Mike did is a great indicator of the kind of scientists that we have at our disposal. These are people that aren't just thinking about it from a scientific perspective, but they want to engage with our community. In fact, that's one of the greatest strengths that has come up again and again and again in discussions of KIF1A.org is that the community gives input, the community interacts with the researchers, and the researchers want to come back for it again and again and again. We've also raised $2.5 million since 2017, and that's incredible. This has been through generous donations and fundraisers by all of you. It's also been due to kind sponsorships from industry and academic partners, private donors. And now we're at the place where we're figuring out, with all of this collected, where do we go? Ooh. And I do just want to, and Jane, you might actually recognize this slide because this is something that I used a lot during graduate school. I do just want to visualize the way that our community has grown from a single individual to over 500. And this image is a misnomer too, because they're all the same color. And what I've found over the last several days is that this is a very vibrant community with unique experiences and skills and talents. And this is something that we want to bring to the forefront. We want to bring in as much of our community as possible and really leverage it to maximum effect. I saw that Angie has been doing, and Angie Fuller is our interim executive director, and she has been doing so much behind the scenes. Angie stepped on over 6 months ago as a volunteer, and she has been doing so much work from the perspective of somebody who is new to the science, but very engaged with the community and trying to bridge those gaps. And so something that I saw Angie doing earlier was she was going around and asking, hey, what is your mutation? Can we connect you with other families that share this mutation? Can we find ways to connect you with one another? Because as much as we love being a hub, we want you all to contribute your direct experiences. We want you to mentor each other. And so this is just a short list of some of the mutations that are present at this conference. And I want to bring to your attention that this is just the tip of the iceberg. We have over 100 mutations represented across our community. And that provides a lot of opportunities for continued conversations and interactions and learning from all of you, because these may be different mutations, and it's important to understand where are the differences, where's the overlap, and how do we come to our common goals. Just another picture of community growth, but I want to give some context to this. Before 2017, we didn't have animal models for CAND. We had a small community that was looking for any representation in science. And in 2017, my predecessors went through something really incredible, which was you all started the We Need a Mouse campaign. And the goal was to have a mouse that represents something about cancer that we can start to do preclinical testing. And with help from Jackson Laboratories, we now have multiple mice. And these mice are being leveraged and used by our research network. In 2019, we needed cells because we wanted to do patient-relevant cell culture experiments for therapeutic development. We have 6 patient-derived cell lines and then 10 genetically engineered cell lines that all represent different aspects of CAND. These are resources that have come because of your engagement and because of your ability to amplify our needs. And so now we're standing at this point where we have grown our community tremendously. We've grown our family community through interfacing with other rare genetic or rare disorders that share diagnosis with CAND. We have reached out to families, and they've reached out to other families, and we've even heard stories of individuals who didn't know that their kid had CAND until they ran into another kid who happened to. And we've also grown up our research network. And the reason that I joined KIF1A.org was something that Kat actually told me when I was interviewing. And she said, Dylan, we've done a lot of growth. We have grown exponentially in the last several years. And what we want to do now is consolidate it. We have built these communities, and now we want to make sure that they are interfacing and interacting in a way that benefits us all. And so that's what I want to talk about. I want to talk a little bit about what that consolidation means and what it means to have all of the pieces of the puzzle interacting with one another. Our general therapeutic strategy, and we talked about this quite a bit yesterday, was to find CAND treatments and cures. And there are 2 ways to go about this. We can go for specific KIF1A targets. These are the ASOs and gene therapies that we've talked about yesterday. And then there are also global targets that are meant to modulate global pathways. Can we stop neurodegeneration? Can we target epilepsy symptoms? Can we repurpose small molecule drugs? And in order to do that, we developed a Treatment Accelerator Program. And the idea was very simple. Can we get neurons with KIF1A mutations that are representative of our community? Can we see how they differ from normal neurons? And then can we start to test compounds in that? And this is part of what NewSite has been doing. It's also something that the MCRI and Dr. Sim has stepped into. And we're now in an abundance of people that are going through the process of looking at these cells. Our next session, or our afternoon session, will consist of 3 different groups that all got mini grants from us to study the patient-derived iPSCs. And so we have group upon group upon group that is now looking at what is it about KIF1A mutations that make neurons behave differently, and can we now use that to screen compounds? And the goal for this is that we now have individuals with different levels of expertise at different stages of clinical development, and they are not only all working on this, but they are working on it together and communicating. And when you have all of these individual types of research and they are all converging on the same goal, when they're all aiming for the same thing, that's how we get patient-centered research. And so it really is a success of our community that we have so many researchers that are thinking about the people in our community as they're going to the bench, as they're working with their mice, as they're looking for new compounds. And what is our role in this? A couple of years ago, this was our vision. It was that KIF1A.org would be a hub, and we had several research partners that were working with us. And the idea is that if we are communicating with them, if we're at the center of this, if we are sharing resources between them, we will begin to develop a treatment accelerator program. And this is still true, but I do want to show something that we worked on a couple weeks ago, or a couple months ago, which was Dom wanted to see, what does our research network actually look like? What is the shape of our research network? Because this doesn't quite cut it. We have too many researchers and they're interacting too much. And so what we wanted to do was make a map. And I have just the shape of it not every single research network member is represented here, and they're not all labeled, but I just want to show you the shape of the number of groups that we have working at every stage of development. And not only the fact that they're working on it, but look at all of the lines between them. These are people that are sharing knowledge, sharing resources, sharing protocols, and coming back to each other with their results so that they can refine these experiments again and again and again. And that's a truly incredible thing. So this is what we mean when we talk about consolidating our network. Not only do we have all of these researchers working, but they're working in tandem. And that's massive. I'd be remiss if I didn't mention that our major goal in 2022, other than consolidating the research, has been the koala study. And in that regard, this has already been a huge success. By the end of yesterday, when the blood draws were finished, we were looking at over 60 patients that are represented in the koala study. That's 1/10 of the patients that we know about. That's huge. And not only that, but we've been able to do it in conjunction with very generous partners like Ovid, who have been able to help families get to the koala study. And so as we move forward, we're looking to obviously expand both the natural history study and the koala study. We want to get more people out here. We want to get more data about this. And this is something that we'll continue to work on and be committed to as we move forward. I also want to celebrate something really important, which is that we are a small nonprofit. And so it's very important that when we invest in things that we make sure that they're sustainable. And one of the greatest things about our research network is they are always looking for ways to take the load off of us. They are always trying to look for ways to get the work done without it being something that we have to worry about. Murdoch Children's Research Institute leveraged our pilot funding, and Dr. Simran Kaur was able to secure $750,000 from the Australian government to study KIF1A. Dr. Hernando Sosa and Dr. Arne Genrich got a $100,000 Exceed Award to study KIF1A structure for the first time, and now we've seen the fruits of that labor. Recently, the Jackson Laboratory was awarded an NIH Ignite grant to expand the types of KIF1A mouse models that we have. So now we have a mouse, we have mice, and now we're getting better and better mouse models that will represent more aspects of CAND. And we'll talk about that more later with Marcus. We're also very blessed because we've had research partners who have started doing the work for us, and because of the opportunities in it, because of their passion for it, have continued to do the work above and beyond. And for that, I really do have to call out NewSite and Dr. Wayne Poon, because every time we go to them, they are doing more experiments than we expect them to, and they are looking to expand the program, and they are always looking for ways to make that something that is sustainable in the long term. And that is not necessarily something that you always find going through this space. These are people that are committed to the long run, committed to helping us find treatments and cures, and doing it in tandem with one another. Yeah. A lot of this was done with seed money, and that seed money came from a very, very incredible, generous gift, which was the Rare as One grant from the Chan Zuckerberg Initiative. This was a grant that was for capacity building. And not only did it provide us money so that we could fund NewSite and support the Chung Lab and support these other projects, as well as provide support to individual families when they needed it, but it also hooked us into the broader research network. We now have dozens of rare disease organizations that are speaking with us regularly, meeting with us in peer support, and looking for ways to form best practices. How can we learn from other rare disease organizations so that we don't have to reinvent the wheel? How do we make sure that we're as successful as possible by banding together when our need is common? And that's something that has been incredibly, incredibly helpful for us, not only in our operationalization, but also just in the way that we think about moving through these spaces. We have people who have gone before us and people that are behind us, and we're all learning from one another. And that kind of a community is invaluable as well. So even as you look around, you know that there are many, many, many other groups that are thinking of you and our group and are looking for ways to improve our lives. So this was a success that I don't know, Luke, you may disagree, but I think unanticipated in 2017. And we now find ourselves amidst so much success that we're looking at how do we expand into this next stage of what we're trying to do. And so I just wanted to point out some opportunities that we see in the future, things that we want to leverage. And I bring this up because we've grown to over 500 families. And that means a lot of expertise, a lot of skills, a lot of perspectives. And so in the year to come, expect to hear more about how you can help and contribute, because we need it. We need all hands on deck, and we want to leverage you in the ways that you find helpful and are passionate about. So amongst those, clinical care guidelines is something that's very near and dear to our heart, because we know that many of you, when you go to clinicians, have to do the educating, and we want to lift that burden off of you. And that's one of the reasons why we've been having these clinical conversations, is we want to start bringing our awareness closer to the clinical manifestations of the disorder so that we can start giving advice, and the Koala study will feed into that, but also soliciting feedback from all of you and starting to talk to more of the individual clinicians that you meet with will be invaluable in the future. So in addition to our research network, we would really like to establish, build, and consolidate a clinical network. Patient advocacy and policy is something that has come up a lot. And I believe Dr. Chung mentioned it, Dr. Poon mentioned it, that there are a lot of opportunities in which advocacy can make it easier to secure funding, can make it easier to get coverage for any kinds of therapeutics that are developed. And this is a space that we haven't traditionally had to step into, or have, with the power of individual personalities, had to forge through, but now we're looking at changing policy. And so to that end, Stephanie Fisher, who has also been doing about 1,000 jobs in the background over the course of the several days, has been meeting with us to suss out some of these things, because she has experience with this kind of patient advocacy and policy. And so that's something that's on our mind and something that we're going to be moving into, and you can expect to hear more from us about what that will mean in the future. Integration of high impact symptoms. This has happened a lot over the last years. We have better mouse models. We have Simran Kaur, who has taken research roundtables and turned them into new projects overnight. And so this is something that we are very excited to say is already happening. And now it comes down to, how do we get all of these various models that integrate different symptoms, and how do we get them into effective pipelines for treatments? So now that we have all the puzzle pieces, how do we start to fit them together? We are operationalizing platforms like NewSite with pharmaceutical developers. We're looking for more ways to get these platforms underway and sustainable and attractive to others, so that we can start to churn potential compounds through, because we've had a lot of success with hypothesis generation. Now it comes to the point where we have to start assessing. And we also want to coordinate with the international groups. Like I said, there are many people across the world that are taking this into their own hands. And it is now coming to a time when we want to leverage the strengths of all of these individual groups and start to integrate an international research community to an even greater extent than we have in the past. And lastly, fundraising is something that we will be thinking about a lot in the year to come. I will leave it relatively loosely at that, but this is something that is important for any kind of a rare disease organization. And we have a lot of expertise within this group and people who have taken on fundraising projects. And this is another great example of where we can learn from our community and we can get best practices and start to put this into something that is more of a sustainable format that we can plug and play and really start to increase the war chest. And as always, I'm a little bit over time, but I do want to end with just one example of our research network at its best. Dr. Shi is a professor at Stanford University, and she was trying to run an undergraduate class on biology, on genetics, with C. elegans. And luckily, we have somebody who is an expert in C. elegans, Dr. Shinsuke Niwa. And so she reached out to him, and she asked, well, how do you think we could use these kinds of KIF1A mutations in a class to teach undergraduates about biology? Dr. Niwa reached out to Dr. Chung, and Dr. Chung connected to KIF1A.org and the families. And one of the things that went into this was having families attend the class. And not only that, but soliciting comments from the community on how, if we had all the money in the world, would we improve the lives of our family members. And that was invaluable because it also meant that we got to see those comments and think about them. And something that came up in that context was vision. Again and again, vision has impacted my quality of life. I don't know how well my child can see. I thought that they were just inattentive until I realized that they actually weren't able to focus. And we had known this, but what we hadn't really thought about was, how does that fit with our preclinical models? Are we actually looking at vision in CAND? So we did a research roundtable about it, and we talked about what is vision loss, what are the mechanisms by which KIF1A mutations might disrupt vision, and how are we modeling this in our preclinical assessments? Dr. Kaur in Australia, so we've now gone from Stanford to Japan to Boston and all the way to Australia. Dr. Kaur watched this presentation, and almost overnight, she reached out to me and she said, hey, can we talk to Jackson Labs because we want to better understand vision in CAND. And Marcus and Dr. Kaur met and they talked about it specifically. They said, how can we do this? And they are now collecting optic nerves from their mouse models and sending it over to Australia. And so this is a great example of how a network that spans teaching and education to preclinical research, clinical research, families, back in feeding to more preclinical models that are going to better represent our community. And then Sim also did something that really surprised me, which she asked me about speech, because we had talked about how vision is very difficult to communicate. And when we brought up that speech is something that we don't know a whole ton about from, the literature, she connected us with Dr. Morgan in Australia to start looking at speech specifically, which she spoke about a little bit. But I just want to point this out as an example of all of the different ways in which our research network can interact. And there's more opportunity for this, and we're going to look for ways to make this an even more regular occurrence for our community. So with that, I think I am over time, but I hope that this gives you some things to think about the way that we can all fit together. And I hope that you expect to hear from us about ways in which you can participate, because one thing that I've learned over the last several days is this is a truly incredible group of people, and it would be a shame not to leverage you all. So we'll be talking more, and please do reach out if you have questions, thoughts, comments, or just want to talk. Thank you very much.